This is the common feature between plastids and mitochondria. Yes, these are the structures in which DNA is present. The first point is also right, that is they have extra nuclear DNA or extra chromosomal DNA. That is mitochondria as well as chloroplast both have double stranded DNA molecule. Then both of them have ribosomes also. DNA is there, ribosome is there. So most of the enzymes and proteins are synthesized by the chloroplast and mitochondria of their need. That's why they are semi-autonomous cell organelle. Because of presence of these two structures, mitochondria and chloroplast are called semi-autonomous structure. Semi-autonomous structure of cell. Semi-autonomous structure of cell. That is chloroplast and mitochondria. And third, both have double membrane. So all these features, that is, all these features are same in both the cell organelle. So that's why answer all of these is correct. All of these is correct. And one more, they self-replicating structure. They divide, they multiply by the fission process. Multiply by the fission process. 37th, number of chloroplast per cell, and that is in the mesophyll cell. That average number, this is data based question and this is the answer. In the mesophyll cell, number of chloroplast 20 to 40. And what is mesophyll cell? That is the tissue present between the two epiderm of leaf. I have already taught in the anatomy section. That is, the tissue present in between the two epiderm of a leaf is known as mesophyll. It is chlorenchymatous. It is chlorenchymatous, that is parenchyma with chloroplast. And the number of chloroplast per cell, number of chloroplast per cell, this is number of chloroplast per cell of mesophyll. Number of chloroplast per cell in mesophyll is 20 to 40. Least number of chloroplast case is chlamydomonas. Chlamydomonas has only one chloroplast. Chlamydomonas has only one chloroplast and that is cup shaped. Only one chloroplast. One chloroplast is found in chlamydomonas. Chlamydomonas has only one chloroplast. That is organelle within organelle. Organelle within organelle. That is, it is a cell organelle, but it is present inside the cell organelle also. So ribosome is the structure. It is a cell organelle found in all cell. Found in all the cell, but it is non-membranous cell organelle. Ribosome is non-membranous cell organelle. That is, there is no membrane. Found in all cell. Even though it is found in two structures also, they are present inside the two structure also. One is mitochondria. One is mitochondria. Second is chloroplast. Second is chloroplast. And the ribosome present in them is 70 S type. And what is S? It's a sedimentation coefficient. That is, it is measured in Swedberg unit. It is measured in Swedberg unit. S is actually written for Swedberg unit. So ribosome is present inside the cell organelle also. That's what is called organelle within organelle. The common organelle present in both the cell, that is again answer is ribosome. That is both cell means that is prokaryotic and eukaryotic cell both. The answer is ribosome. It is a non-membranous cell organelle, was reported by Pallade, P-A-L-A-D-E. So they are also called Pallade granules. These are also called Pallade granules. These are also known as Pallade granules. So prokaryotic and eukaryotic cell, because in prokaryotic cell membrane bond cell organelles are not found, and this cell organelle is non-membranous cell organelle. Ribosome is non-membranous cell organelle. Centrosome is also non-membranous, but it is found in the animal cell only. Even plant cell it is not found. Found in animal cell. Non-membranous cell organelle, centrosome, is found in animal cell. Cell membrane is a covering found in all cell. Vacuole, as far as large central vacuole, it is found in the eukaryotic plant cell only. But for this question the answer is 3. Cytoskeleton. Cytoskeleton, for the formation of cytoskeleton all 3 are required. That is microtubule, microfilament and intermediate filament. All 3 are the part of cytoskeleton. These are all the part of cytoskeleton, and they are the special proteinous structure. Microtubules hollow. Microtubules are hollow and made up of tubulin protein. Whereas microfilament, solid filaments, these are solid filaments made up of myosin actin protein like structures. And intermediate filaments, they are also proteinous, they are called tonofilaments. These are known as tonofilaments. But here look at the question, that is out of these 3, which one is helpful in protoplasmic streaming? The answer is microfilament. Microtubules actually structural part of cilia, flagella, basal granule, spindle apparatus. They are the structural part of cilia. Number 2 flagella. Number 3 basal body, through which cilia or flagella originate. Number 4 they are the structural part of centriole. And number 5 they are the structural part of spindle fibers. This is about the microtubule. So here the answer is 2. That is, they are responsible for the cytoplasmic streaming. The microtubule constitute, I have already given the idea, spindle fiber, centriole, cilia, right. This is the correct answer, spindle fiber, centriole and the cilia. Next, main function of nucleolus. This is nucleus and its membrane is porous and double. Then there is a non-membranous structure. This is non-membranous structure found inside the nucleus, that is known as nucleolus. This structure is called nucleolus. It is a non-membranous cell organelle. And nucleolus, it has 2 major roles. Number one, rRNA synthesis, that is ribosomal RNA synthesis. Second, ribosome biogenesis. Second major role is ribosome biogenesis. Two major roles are there, 1 and 2. So the main function of the nucleolus, that is ribosome biogenesis, but in this case answer is rRNA synthesis, ribosomal RNA. Chromosome. The chromosome which we see in prophase stage, it has two chromatids. And these two chromatids held together a structure known as primary constriction or centromere. This structure is centromere. And these are the two chromatids. On centromere, there is a disc-like structure that is known as kinetochore. This is a disc-like structure over centromere. This is known as kinetochore. Proteinous disc-like structure known as kinetochore. Clear? And it is a site of attachment of spindle fiber. And these are the ends of chromosome. Ends of chromosomes are called telomere. They are known as telomere. The ends of chromosomes are called telomere. And this is centromere. And on which this structure is kinetochore. Capsomere is related with the viruses. Chromomeres are the units in chromatin, color bearing bodies. So ends of chromosome are called what? Telomere. It has great role, that is the structure and stability of chromosome. Telomere is concerned with the structure and stability of a chromosome. What is the role of telomere in chromosome? That is, they are concerned with the structure and stability of chromosome. And chromosome can also be classified on the basis of position of centromere. So let's know, if centromere is in the center, this is metacentric chromosome. If centromere is closer to center, submetacentric. If centromere is closer to end, this is acrocentric. If centromere is at the one end, then it is telocentric. The 4 different types of chromosomes are there on the basis of position of centromere. You can also display by them in 2 arms. This is centromere. Right? And this is very short, almost at the end. Then it is telocentric. So 4 types of chromosome. First, metacentric. Centromere is the median position. Second, close to center, then submetacentric. Then close to end, then acrocentric. If they are present at end, then telocentric. These are the 4 different types of chromosomes. They are already given in the NCERT book also. Now come to the Position based question. Position of centromere is subterminal. Answer very simple, that is acrocentric, close to the tip, not at the tip. Here at the tip, here close to center, and the last one is in the center. Then chromatin. This is nucleus. This is nucleus. Within nucleus, we have already described, this is nucleolus. This is nucleolus, non-membranous structure. And within nucleus, within the nucleus, nucleoproteinous substance is present with the affinity towards basic dye. This is nucleoproteinous substance found in the nucleus. This has the affinity towards basic dye. This is actually chromatin. This is chromatin. This is chromatin. And if we analyze chromatin, the amount of DNA is 40% in chromatin. RNA is very less, 1.5% only. Highest histone protein, that is basic protein histone, 50%, and NHC, non-histone chromosomal protein, NHCP, 8.5%. So, chromatin is basically rich in DNA and histone. These two are 90%. DNA and histone. But we can't ignore the presence of RNA as well as NHCP. So, my answer for this question is DNA, RNA and protein. Protein covers both histone and non-histone. Protein covers both histone and non-histone. And this chromatin undergoes condensation and dehydration during the cell division. This chromatin undergoes condensation and dehydration during cell division and appears in the number of thread-like structures. They are in definite number. They are called chromosome. So, chromatin results in the formation of chromosome, but the chromosomes are seen only at the time of cell division. They are visible in prophase and they disappear during the last phase, that is telophase. Appear in prophase, disappear during telophase. Similarly, we can say chromatin condensation takes place in prophase and decondensation takes place in telophase stage. This is all about the few questions of cytology. Thank you.